If you can make it to these, great. If not, that's okay. I will record them and post them for you so you have access to those and you can rewatch them or whatever. So this is really the only day where I will go through the lab. Like if there's like a PowerPoint for it. The rest of them, it will be, you know, a 10-minute explanation. Here's how we're going to do this. I also posted for you a little video that demoed how to use some of your equipment. So if you know, the reflex hammer, that's an easy thing. You'll be able to figure that out. But I did demo things just in case you were, you know, unfamiliar with that. I'm also going to do a demo. I'll make one today of how to use the blood pressure cuffs during exercise. So I needed a partner to do that, so I couldn't do that on Friday. But I'll make sure that to suck in one of my colleagues in order to do that for you guys today, before I give you all the blood pressure cuffs, because I'm giving you everything I have. So I'll get that done this morning then before I send everything out for you. Any questions on anything so far? Everybody's good. Okay. How many of you have taken either our research methods class or a research class, maybe in psychology or a different department?
Yeah.
Okay, so Val's the only one. Okay, cool. So this this first lab is kind of an intro to the scientific method. So for all of you, probably except Val, this will be maybe new information. Okay, you can see my screen?
Yep.
Okay. All right. So again, in real life, our lab session format, on those Monday, Wednesdays, and Fridays, I'll explain to you guys what you'll be doing in those labs. I have a Google Doc set up for each lab where you'll need to share information, where you need like the class data. So I didn't put a due date for each of those, but just try to stay on track with the schedules. I know I'm not setting you up for success right away because you're not going to have some of your equipment right away, but for example, you know, we've got the sensory lab on Wednesday. Okay, so if you can get your class results posted by like Wednesday night or Thursday morning, then you have a sensory presentation that you'll do on those results that following Monday. So try to get those results posted as soon as you can. That way, you know, if some of you, I know some of you, a lot of you have jobs and other things going on, so you can kind of stay on top of those assignments because you'll need some of that class data. Okay, there's multiple stations for each lab. Again, if you've looked through that, everything that you need is already posted for the semester. So there are multiple activities and things like that. Some activities everyone will complete, and some, you know, we selected a couple different participants to complete those. Okay. For each lab, there is some sort of assignment, whether it's a presentation, a lab report, a couple questions to answer, or just a handout. Okay. I'm going to talk in a lot more detail about the lab reports when we get to them. I believe our first lab report is next week. So I'll probably spend a lot of time on either Monday or Wednesday of next week talking about how to organize those. I also posted an example lab report for you so that you can look at how the formatting is and that sort of thing. How many of you are familiar with like APA formatting? Yes, yes, yes. Okay. All right. So I would prefer that we use APA formatting. That's like the scientific reference standards. So again, I posted an example of how that looks, but I'm happy to give you guys more guidance. And like I said, I'll go over that in a lot more detail maybe next week when we're starting to get ready for that first lab report. You'll hand those in via, what's it called, turnitin.com. So there's a Dropbox for each of those on our eLearn site. And basically with turnitin.com, it'll check to make sure that you haven't plagiarized with, you know, something that's already published online. but it'll also check that with other Loris students and other colleges that use turnitin.com. Okay. So my point is that you can't take, your friend that took this class last year, don't take her lab report and change it and hand it in. Okay. Because it'll show up as plagiarized and then you fail the class. So don't do that. So again, all of those things are checked against Loris students and against anything that's out there on the internet. So that's kind of how turnitin.com works. Not that I'm worried about this group. I think we have a really good group. This shows you how to do that APA formatting. And again, I'll go over that next week in a little bit more detail. Okay, so citation. So I can't remember if it's the first assignment or I think it's the second assignment. So when you do your nervous system assignment, it does ask you to cite some things. Okay, so when you use APA formatting, notice that if I'm going to write one paragraph, then I need to cite at the beginning of the paragraph and at the end of the paragraph. And then that assumes that everything in this middle section came from that same source. Okay, so do try to cite at the beginning and end of each paragraph when needed. And again, use that APA reference form to, cite your sources at the end of the or at the end of your assignment as well. In some of your lab reports and things like that, again, blood pressure and cardiovascular system is the first lab report that you write. Make sure you label all your axes. So again, here we're looking at the difference between systolic blood pressure and diastolic pressure for males, females versus the whole class. I want to make sure I label my y-axis as well. So blood pressure is measured in millimeters of mercury, and so that can go on this axis. So again, that's a little bit of information about formatting and things like that. Certainly if you have questions as you're working on these, you can, if you have the free time and you want to get ahead on stuff, go ahead. Everything is ready to go and posted for you. You should be able to, if you want to get ahead on the labs, go for it. If you want to get ahead on the lectures, again, everything is completely ready for you. The only thing that you'll have to wait for are those quizzes or exams. But otherwise, if you wanted to hand stuff in early, feel free. Or you want me to look over, I'm not sure if I cited this right or whatever, just email me a draft and I'm happy to look over those types of things as well. Okay. So again, in the first half of this class, we focused a lot on the anatomy, which was how things are organized and what the structure is. And now in this portion of the class, we'll focus more on the physiology. Okay, so how do things work and how does the structure of those systems play into what the function is? Okay, so again, I like this section because it's a lot more applied and you get to think about why things happen. And so I enjoy that. Again, we'll be pulling from a variety of fields. So anatomy and phys is a blend of biochemistry, which looks at, energy metabolism. So being able to convert molecules into different forms. Physics, what are physics doing in here? What do physics have to do with this? Anybody take high school physics at least? Come on, what are physics doing? How are they related to how the body works?
It's kind of like how the laws of gravity and what we have on Earth apply to the human body.
Sure. How many of you have taken biomechanics? Nobody? Val, you're the only one, right? Yeah. So biomechanics is basically the physics of the body. You can think of it that way. And again, I'm a huge I don't care about health that much. I know that sounds weird. I don't care about health at all, actually. I just care about sports performance. And so when we think about physics and the application of that to the body, how can I manipulate my levers so that I can throw farther or more accurately, right? So again, thinking about those components when we think about body movement, I think is pretty cool. How do we use math and physiology? How might math be important?
Like to calculate like the velocity of things and like blood, like how much blood there is.
Sure. And again, when we think about science and experiments and things like that, we use math or statistics in order for us to figure out, well, was there a difference between these two things? Okay. So without statistics, there isn't really a way for us to determine whether or not this difference might be significant or not. Okay. Biology, again, looking at things at the microscopic level is important as well, so that we can maybe justify or understand why things are happening the way they're happening, things like that. So again, the idea is that this isn't like a separate thing, right? We're applying all these different pieces of maybe what you've already learned in order for us to make sense of how the body is working. Okay, so again, when we think about science and the scientific method and experiments and things like that, there's two variables, okay? So we have an independent variable, which is something that we control or manipulate, and then we also have a dependent variable, which is something that we want to measure. So I want you guys to think for a second, come up with a question, something that you're interested in knowing about the human body. Again, it can be related to injury, disease, sports performance. Throw something out there. What's something you're curious about in the world of kinesiology or whatever your field is that you want to go into? Or throw something out about COVID. Just throw me a question. Anything you're curious. I hope you're curious about something. Jess, what do you got?
How does altitude affect your VO2 max?
Okay, so we want to understand if altitude, like altitude training, is that what you mean?
Yeah.
Okay, so is altitude training going to be beneficial for your VO2 max or your aerobic fitness, right? So one variable, and again, this is kind of weird to think about it this way. One variable we're manipulating and one we want to measure, okay? So again, if I wanted to set up this experiment, what might I do? How could I determine whether altitude was helping my VO2 max or not? Can you throw out some ideas about what type of experiment or what could we do?
Have athletes train at different altitudes for different amounts of time and then have them like flip-flop where they're at, like high or low.
Okay, and then measure their VO2 max in either scenario, right? Okay, so what is the thing that we're manipulating or controlling?
Altitude.
Yep, whether or not they're at altitude. Okay, and then the dependent variable, what are we measuring?
VO2 max.
Yep. Okay. So again, it's weird to think that we're manipulating altitude, but in reality, that's what, that's how we would set up that experiment. So the independent variable was which environmental setting they're in, and then the dependent variable was what was their aerobic fitness based on which scenario that they were in. Okay. So the independent variable is represented on the x-axis, and the dependent variable on the y-axis. Okay. So here you can see something that I'm pretty interested in is the lactate threshold. And so here you can see lactate is charted here as it relates to time on a treadmill, okay? And so essentially what we're doing in this experiment is every time stage, the intensity on the treadmill gets harder, okay? That's what a graded treadmill test is. This is like an actual experiment that I have done before. Okay, so like here you're starting at a light jog and then it gets a little bit harder and then it gets a little bit harder, etc. Okay, and so what I'm looking for is the change in lactate as time progresses, right? So which of these is the independent variable? What am I controlling?
Time.
Yeah, it's super weird to think that you're in control of time, but that's really what it is, because this test is graded. Okay, so time is the variable that is independent that we are manipulating, and then the dependent variable, what do we want to measure, would be
Lactate.
lactate, right? We want to see how lactate changes over time. Okay, so that would be our dependent variable as charted here. Again, make sure you would label each axes. Axes, is that the right way to say that? I don't know. I think it is. These I think you probably learned in like 6th grade, but just in case you didn't or you forgot, I figured I should cover my bases. When we do experiments, again, it's important for us to have some sort of standard or way to determine whether or not there was a difference based on whatever that experimental protocol was, okay? So we have the mean, mode, median, range, okay? So the mean is just your average. So it's the sum of your numbers divided by how many numbers you have, okay? The mode, how often a number occurs. So again, the number that occurs most often in this data set is 12. There's 3 12s there. And then the median, I'm going to put all my numbers in sequence, okay, and figure out what's the middle number. Can you think of a reason why I might use the median versus the mean?
It's resistant to outliers.
Yep, so in some scenarios, like let's say there was like a 26 on this data set, for example, okay? Well, that 26 would really bring my mean up. Okay, so using a median in that scenario might be better because it kind of takes care of that outlier a little bit. Okay, same is true if I had like a 1 or a -3 or something on this side. The median might be better because it kind of accounts for that one number or that one, variable that was out of whack a little bit. Okay, range gives us information about how big your data set is. So it just gives us more information about what's going on within that experiment so that we know the difference between your highest and your lowest number. Okay, so I do want to spend kind of a significant amount of time on this. So this will give you some information, so make sure you're taking some notes here about which statistical analysis you're going to use depending on how we set up the experiment. Now, when you take a research methods class, which I would assume most of you will, Janae, I don't know if that's part of your requirements for your program, but it is for us at Loris. It's important for us to kind of have an idea of which statistical analysis we want to run. Okay, so I'm going to give you an example of each of these. Again, this is pretty simplistic. When you would take that research methods class, we'll get into some more options. There's a lot more options, but these kind of cover our general bases. Okay, so if I want to do a t-test, remember that there's 2 of something. Okay, and with an ANOVA, there's 3 or more. Okay, so a t-test, I can have 2 time points, which is a paired t-test, or I can have 2 groups, which is an unpaired t-test, okay? So a paired t-test, I have 2 time points. I wanted to see what your amount of sleep was that you were getting each night while you were in school versus how much sleep you're getting each night during the summer, okay? So 2 different time points, but the same group of people. Okay, so I'm going to take everybody in this class or everybody at Loris College or whatever, okay, and measure them in 2 different time points. We would use a paired t-test in order to determine if that was a significant difference in the amount of time you slept. Okay, unpaired t-test takes 2 different groups. Okay, so an unpaired t-test, maybe I wanted to know, is the vertical jump of our women's basketball team different than the vertical jump of our women's volleyball team? Okay, so 2 different groups. Now we have to assume that there's no girls that are on both teams. Okay, so 2 separate groups, 1 time point. We could test all those girls at the same time on the same day. Okay, so those are your 2 different types of t-tests. Okay, again, with an ANOVA, you need 3 or more. So with repeated measures ANOVA, maybe I want to look at how much sleep you're getting in 3 different scenarios. So maybe we want to look at the amount of sleep you're getting during your spring semester, during the summer, and during the fall semester. Okay, so there are 3 different time points, but I'm looking at the same group of people in each scenario. Okay, single factor ANOVA is 3 or more groups. Okay, so with single factor ANOVA, maybe I want to look at vertical jump for women's basketball, women's volleyball, and women's soccer and softball. Okay, so I have 4 different groups. They're all mutually exclusive. So we would have to assume that none of those girls played on more than one team. Okay, so as long as we could assume that those groups were all separate, okay, we could test all of those girls, all those groups in one day. Okay, so that's single factor ANOVA. Okay, now correlation and regression, these get mixed up a lot. Okay, so with a regression, I want to understand if there's well, let's start with correlation actually. With correlation, I want to know if there's a relationship between 2 variables, okay? But it's not causal. So let's see. A good example of correlation might be, I want to know if there's a relationship between the amount of money I make, so like my salary, and my level of happiness, okay? So let's say we had some sort of survey that figured out how happy I was on a scale of like 1 to 100, okay? And then you took how much money that individual made as their yearly salary, okay? Now, what I mean when I say it's not causal, okay? This is a philosophical question, I think, but was I happier because I was making more money? Or was I making more money because I was happier, right? So we can't say that one of those variables causes the other one to happen. We just are simply looking to observe and determine whether there's a relationship or not between those 2 things. So that's what correlation is, okay? And then with regression, we do have a clear independent and dependent variable. We want to see the relationship on a continuum with that. So pretty much all the research that I do is a regression. Okay, so I like regressions because we can use this information then to predict what might happen. Okay, so let me think of a good regression example. I'll just use some things that I've done in my research. So one variable that was interesting for me to look at was body fat storage, like where you store your body fat and how that relates to your 5K time. So I took a group of like 35 highly trained female athletes, female distance runners, and I measured their body fat percentage in their like abdominal region. Okay. And so again, we wanted to look to see if there was a causal relationship. Could I determine or predict what their 5K time might be just simply based on how much fat they had stored in their midsection? Okay, so that's an example of regression. So the independent variable was body fat percentage, and then we wanted to know how that affected their 5K time, which was their dependent variable, or what we measured, if that makes sense. Yes? Everybody, how you doing out there? How many people do we have on board today? We got 1, 2, 3, 4. Okay, so what I want to do now is you guys work together. I'll have you work in one group, and I want you to come up with a different research example for each of these kind of categories. Okay, so come up with a regression, a correlation, a paired t-test, unpaired, repeated measures ANOVA, and single factor ANOVA. Okay, do you need me to leave the PowerPoint up, or do you have your notes pulled up? Or one of you could share your screen and you could like write them out. So many options here. Let's see. I have it set so one of you can share your screen if you want, but work together to come up with each of those categories, okay? Ready, go.
All right, who wants to share their screen?
I can try to go pull it up. I don't have it downloaded yet, though.
You could just do like a Word document too, and then just kind of list out I can put them in the chat box too, if is that helpful? If I put each of those categories in the chat box?
Yeah.
Okay. There you go.
Okay. Am I sharing the screen?
Yeah.
Okay. So you guys want to start with regression? Or do you want to
Sure.
start with that? Okay. So what are we doing again? We're just making up like asking a question.
Yeah.
Yeah, we're putting it in an
Examples.
example.
So my stats class, a big example that we used for this one was like the number of TVs and the number of cars you have, or like, because like if you have more money, you're probably gonna have more of both, but it doesn't mean that you have car, like you're gonna have a TV that comes with it. Just like usually if you have more money, you have more cars and you have more TVs.
Okay. Should I just write that or should I write more?
Jess, are you sure that's for regression or is that for correlation?
Wouldn't that one be for correlation and regression more so like, I don't know, like how much water you have before like a game, do that affect like your performance or something? I don't know.
Oh, sure. Which one?
Or number of TVs and cars would be for correlation. And then regression would be, what did I say? The amount of water intake before a game have like, what did I say? Have an effect on the way you play or performance.
Does it like perform, like your performance
Mm-hmm.
in something? Okay. So is that good for those two? Okay. And then
Okay, good t-test.
oh, the paired one is like if like we're taking a test with like the same people, right? So
Mm-hmm.
At 2 different time points.
am I supposed to just write like what it is or like an example of what it is?
An example.
Okay. So um I don't know, like the caffeine lab, like isn't it like everybody's getting tested or whatever in our class, right? Is that something
Oh
that could be
yeah,
paired?
I think that does work.
Okay. And then
You could kind of say the same thing for that one, but have two groups instead of one with the caffeine and one without, but not at the same. Well, I guess they could be at the same time. Definitely.
oh, I couldn't hear that, so I don't know what to write.
Sorry. I said you could kind of do the same thing for unpaired and paired, but instead of have everyone taking the caffeine pill, just have two groups, one with caffeine and one without.
Okay. Okay. The next one is so what should we do for these last two? These are the ones that are grouped in 3, right?
Mm-hmm.
Okay. So
For like repeated measures, could we do like how much caffeine students drink in the morning, afternoon, and night? Would that work?
I feel like it because it's like 3 different sections.
Yeah, 3 different scenarios for like one group.
Or like rather than a person, could it be like grade level, like freshman, sophomore, junior, senior? I mean,
Mm-hmm.
Or should I say for the person?
it has to be like 3 or more groups, but I guess.
Well, the age groups that would go for the simple factor, I think, when we do freshman, sophomore, junior, senior.
Okay. So I should say that.
Yeah,
Okay.
for the simple factor on.
Okay. And then single factor. Um,
It's kind of me how like caffeine affects a person's like sleep, like person's like sleep, glucose levels, and blood pressure.
Yeah.
their sleep, glucose
And
level.
and blood
What was
pressure.
that? Okay. Oh my gosh. Okay.
Okay, so let's take a look at these. Janae, leave them pulled up here. Regression, this one looks great. Amount of water you can take in. Again, one thing that's important with a regression is it has to be on a continuum. Okay, so I could drink 2 ounces of water, I could drink 6 ounces, I could drink 14 or whatever. So it's on a continuum, if that makes sense. The one thing that is super common is you have this thing performance, right? Well, how do I measure performance? How can you make this a measurable variable?
You could measure VO2 max.
Sure. Okay. So we can look at VO2 max. What's another example of a way we could measure performance?
Several time trials.
Sure. Okay. What was their time? And maybe we had them run 2 miles as hard as they can. Okay. Other ways we can measure performance. You know, if we wanted to look at their performance, like in the weight room, what was their maximum number of push-ups they could do, or something along those lines, or what was their 1 RM on the squat. Okay, so again, when we say performance, just make sure that this is testable when you set this up. Okay, and again, that can be a 1,000 different things. There's lots of different ways that we could quantify performance. Correlation, this looks great. Number of TVs versus number of cars. Again, one doesn't necessarily cause the other. We're just simply observing to see if there's a relationship. Okay. Our caffeine lab will actually be a repeated measures ANOVA because we'll test them at, I think it's 4 different time points, but it could definitely be a paired test as well. Okay. So tell me what's the independent and the dependent variable with this experiment that you have set up here for the paired t-test.
You could be measuring like test scores or something.
Okay. Throw me an example of a test score, like
So like you could take a test with caffeine and then take it out.
okay.
Have the same group do it.
Sure. Okay. So we could do like a typing test, for example, with caffeine, without caffeine. Okay. And then unpaired, so same thing with your unpaired, right? So again, you know, you have one group that takes caffeine, one that doesn't. We measure their typing speed for both groups, one time point, and see if there's a difference. Sure. Okay. Repeated measures ANOVA. I am. What's our dependent variable? It looks like you have some independent variables, but what's our dependent variable? What do we want to measure?
Could measure sleep.
Okay. So we quantify if people drink caffeine in the morning, the afternoon, or evening, and then relate that to how much sleep they get at night or something like that.
Yeah.
Okay. The way in which having these different groups would work, freshman, sophomore, junior, would be we have to track those same people. Okay. So maybe we wanted to see with like what your age group is and how that relates to your GPA, for example. Okay. Well, I have to go with one group and look at what's their GPA as freshmen, what's their GPA as sophomores, what's their GPA as juniors. Okay. So again, when you look at that one could be set up as repeated measures or single factor, depending on how you set up your experiment. Okay. The other way you could do that would be to take everybody at Loras College in 2020, group them as freshmen, sophomores, juniors, and see what the GPA differences are, you know, based on that grade. So that one can really be set up in 2 different ways. So that's neat that you guys did that. There is some, you know, flexibility with how you set up experiments. All right, single factor ANOVA. Tell me what your independent variable is.
Caffeine.
Okay, what's your dependent variable?
Sleep, glucose levels, and blood pressure.
Okay, so here you have 3 dependent variables and 1 independent variable. Could you change that one so that you have 3 groups versus 3 dependent variables? Does that make sense?
How a person's sleep, glucose levels, and blood pressure affects how much caffeine they take in. Is that a way to change it? Okay.
Yeah, you guys are, this study is very complicated because these are three separate things. Just use one of these. Use sleep, glucose, or blood pressure. You choose one of those things.
Let's just do the sleep.
Okay, so sleep and caffeine. All right, so now how could I set up at least 3 groups that are separate groups?
Middle schoolers, high schoolers, and college students, and how much caffeine they have affects their sleep.
Sure, there you go. Okay, so again, they're 3 mutually exclusive groups. You can't be in both middle school and high school at the same time, right? So again, you had 3 different kind of age categories and looking at their sleep and caffeine or whatever. Okay, good. Questions on these kind of setups? So essentially what you'll be doing for the rest of the semester is applying these to the experiments that we have in the class in order to know which statistics is the correct one to run, okay? Again, I appreciate that this is not a statistics or a research methods class. So if you're like, you know, I'm not sure if it's repeated measures or single factor, and I want to make sure I'm doing it right, just ask. Just say, like, I think that's this, can you verify that? And in some of them, I tell you which statistics to run, okay? So that's my story on that. All right, let's get back to here. Okay. Other kind of factors to think about with scientific method, creating a hypothesis. So what do we expect to happen, basically? Okay. It's important that this is testable. And again, we kind of already applied this to your example. So when I say, you know, how much water I drink and how that affects performance, make sure that we find a way to make it testable. Okay. And so in your lab reports, you're going to be asked to provide a hypothesis. Make sure that you make it testable. Okay, so again, we'll look at blood pressure changes associated with a couple different types of exercise. Okay, so you need to be specific about what you mean when you say exercise. Is it aerobic exercise that you're measuring? Is it resistance training? So make sure that whatever you set up, somebody else could take that hypothesis and do an experiment to test whether or not that, you know, it was going to work or not, okay? Null hypothesis, I don't really care about that, but I want you to be exposed to it. It's a statement of default where you say there's no relationship between the 2 variables. So again, there's no relationship between caffeine intake and amount of sleep you get or whatever, okay? Controls. So having control in our research is really important in order for us to get some accurate results. So a control condition is a standard against which other conditions can be compared in a scientific experiment. Okay. Can you think of it? Let's think of an example of a research question or study that we could set up where we need to have a research control condition. Let's go with this. So you wanted to know if your typing speed is affected by caffeine consumption, okay? Is there a way you could have a control condition?
I think every time you do the typing test, you do it in like the kitchen or something. Is that?
That is a type of control, but I'm looking for like a testing scenario where I create a control condition.
Is it like you could set up the test, like the typing, like test or whatever, like whatever they're gonna type or whatever, or like the site they go on to practice the typing?
Like making sure everybody takes the same amount of caffeine.
Okay, yes, those are all good. I don't think I'm asking this the way I want to ask it. What if like I tested all you guys for typing and you had caffeine and like all of you were like superior typers? Well, maybe you were superior without caffeine, right? Like, how do we know that caffeine is the reason that you have good typing speed?
do like the amount of time that we have to type it, I guess. You
Okay.
can say so.
So we're going to keep the amount of time the same. Yes, you guys are giving me a lot of good control things.
Like do one without caffeine.
Yes, that's the control condition. Okay, so I have to test you without caffeine to know if caffeine's the reason you're doing better. Maybe you just like, are a super good typer no matter what, right? So in order for us to know that the reason that you're good at typing is because of the caffeine, well, we have to know what you can do without caffeine. Okay, so a control condition is kind of serves as a standard to which we can compare that treatment. Okay, we use control conditions a lot, like with drugs or like, I'm trying to think of another example, like testing the effectiveness of certain products. That was super vague. So like some of the research that I like to do is on recovery. So like, for example, you've seen those like compression boots before. Have you guys seen those before? They look like big snow pants basically, and you put them on and they like tighten and they like, it's super nice and awesome. Do you know what I'm talking about?
Yeah.
Okay. Another product that I've used is like those percussion massage guns, like the Theragun that sounds like a drill, and we like hold it on you and it like feels awesome. Val, you were in that study, weren't you? Yeah.
Yeah.
Okay. So again, I wanted to know if either of those products are going to help us recover faster from exercise. Okay. Well, what if you just recover fast from exercise without doing anything, right? So again, we have to provide a control condition so that we know that they're recovering faster because they're using the Theragun, not just because they're like super athletes and they're able to recover quickly, right? So those are two examples of when we would use a control condition. Again, it will help us to produce the most accurate results and ensure that what we see changes in that dependent variable are actually due to that treatment. So it's actually because they took the caffeine, or it's actually because they used that Theragun or whatever it might be, or it's actually because they took this weight loss supplement or something along those lines, okay? These are the controls you were providing me. Okay, so again, we had them, you wanted to take the typing test in the same location. We wanted to give them the same amount of time, right? Let's think about some of the characteristics of our subjects. So let me go back to that example of recovery. Okay, so I'm using products that are very specific to sports performance and they're marketed to athletes, right? Well, if I took like my mom who doesn't do any, she walks like 20 minutes a day. That's her exercise. Nothing against you, mom. That's fine. But like she's like 60 years old and she walks 20 minutes a day. Okay. If I took her and like all of her friends and tested them on with the Theraguns, like would that give us any type of good information? Probably not. I mean, who cares if you can recover because you're not the one that is going to utilize this product, right? When you think about appropriate subjects, we've got to think about, what it is that we want to find, right? So again, you guys talked about, fluid or water consumption and performance like VO2 max or whatever, okay? Well, using a football team isn't really that valuable for appropriate subjects because they don't care what their VO2 max is, right? That doesn't help them in football, okay? So choosing appropriate subjects is important, okay? One of you mentioned that we should give everyone the same amount of caffeine, and that could be true, but we might also want to consider the size of each individual, okay? If I gave Sia caffeine, for example, and she's a tiny little cross country runner versus some big football guy, if I give them the same amount of caffeine, is the effect going to be the same? What do you think?
No.
Right. So we need to appreciate that body size might play a role in what the dosage is. Okay. Age, gender would also play a role as well. Design of the experiment. So we can do a control group, which we talked about. So having them do, the typing test without caffeine and then having them do the typing test with caffeine. We can also do a placebo group. Okay. We won't do this in this class, but it's something that I do in like sports nutrition a lot. What's a placebo group?
Like they think they're taking the drug, but they're not.
Yep. So they think they're taking the drug, but they're not. So again, instead of giving you a caffeine pill and you believe caffeine is going to improve your performance, maybe I just give you a little like sugar pill or whatever. Okay. So that's what a placebo group is. What about crossover design? Does anybody know what that means? Anybody? So let's say I wanted to test this new energy drink, okay? And I'm going to take everybody in this class, okay? And again, I know most of you as it relates to like whether you're athletes and that sort of thing or not. Let's say I take all of you in this class. I want to know if this energy drink is going to enhance your performance. Okay, I'm going to have you guys do a pro agility test. How many of you know what a pro agility test is? You might have done it in like PE class. Jess, you know what a pro agility is?
No.
Really? Oh. Okay, so
Maybe if you said it, but like off the top of my head, I don't know.
Sarah?
I can't think of it. I may have done it.
Okay. So I start, these are cones. These little 3 dashes are cones. Okay, I'm going to start in the middle. I'm going to run as fast as I can to this cone. Then I'm going to turn. I'm going to run as fast as I can back to this cone, which disappeared.
We can't see your screen.
What the heck?
I think
Or
if
I
you go to
can't at
view
least.
options at the top.
Have you been not seeing my screen this whole time?
Yeah, you have
Yeah.
to click like options and back to.
Can you see me now?
No, it says you're viewing Janae's screen.
If you go to view options, you can click like which one.
Oh, okay, I got it now.
Can you see me now?
Yeah.
Okay, let me start over. Okay, so I have 3 cones. Those are those red dashes. I'm going to start at the middle cone. I'm going to run over here. Then I'm going to run over to this cone. And then I'm going to run back to the center.
Oh yeah, I know what that is. I did that in my workout yesterday.
Yeah.
5, 10, 5s, yeah.
Yeah. Oh, okay. That's what you call it. Okay. So
Yeah.
let's say I wanted to test everybody on this experiment. Okay. Val, have you ever done this test before?
I don't think so.
Janae?
I can't figure out how to see your screen. Okay, now I see it.
Okay.
I had to stop the sharing, the screen sharing on my screen.
All right.
Okay. Okay. So some of you have done this and some of you haven't, correct? That's my point here. I'm not taking an inventory of who has and who hasn't. That doesn't really matter to me. The point is like, okay, well, Jess, you've done this one a lot, right?
Yeah. Yeah.
Val, you've never done it before. Val, do you think by default you would do better at it the second time you try it?
Probably not. Oh, probably. I was like comparing to Jess, but
You personally will get better the second time you do it, correct?
Yeah.
Because you've never done it before, right? Jess is going to be a little harder for her to see improvements because she's already familiar with the test, right? Okay. So if I don't do a crossover design, okay, a lot of people are going to be better the second time just simply because they've now been exposed to the test, okay? Especially someone like Val who's never done this one before. So if I use a crossover design, that helps to control for that, okay? So maybe I have half of the group use the energy drink and do the test. The other half do the test without the energy drink. Then I'm going to swap, okay? And basically what this is going to help with is it's going to account for the learning curve, okay? Because a lot of people will get better the second time they do it. So we want to make sure their second trial isn't faster just because they learned the skill. It's actually because they took that energy drink or whatever. Does that make sense?
Mm-hmm.
Yes? Is everybody okay with crossover design?
Mm-hmm.
You're going to split your group into 2 and then have them do the one thing and then switch treatments and have them do it again. Okay. Standardization of procedures. Okay. So Again, as I told you, like I've used these compression boots to measure recovery in athletes, and that's like a big thing for me, right? I want the compression boots to help athletes because I believe in this product. The company is giving me all this free stuff so that I can show and publish this research that shows that this product is helpful for athletes, right? So let's say I come in to the lab, right? And Val's going to run on the treadmill after she uses the compression boots. And I want those boots to help her. Okay, so she's running on the treadmill and I'm like, come on, Val, you got this. Keep going, right? The next time she comes in, she didn't wear the compression boots. So I don't want her to do as well on this trial. Okay, so now she comes in and I don't say anything. I just stand there, right? Is it possible that how I treated the subject might make a difference in how long Val could run on that treadmill?
Yeah.
Okay. So again, I didn't standardize my procedures, right? I treated her differently in each trial. And so therefore it's possible that that might have an effect on her results, right? So we want to standardize our procedures so that everyone is treated the same and everything is done in the same way for each of those trials. Okay. Calibrate equipment and then reproducibility of results is another way that we can help with control. So a couple terms to kind of go along with that. Validity. Did the test measure what it's supposed to measure? Okay. Reliability is the reproducibility or consistency of the results over several trials. And objectivity is kind of what I just described, is high reliability with different researchers. Okay, so let's start with validity. Did the test measure what it was supposed to measure? Okay. So let's say I wanted to know if caffeine was going to affect my agility. Okay, so I was going to have everybody take caffeine and then I'm going to test them for something, right? In order for this to be a valid experiment, I have to do a test that actually measures agility. Okay, so if I had my subjects come in and do like a 40-yard dash, that doesn't really do it for me, right? That wasn't a good choice for a test. 40-yard dash tells me how fast they can run, but doesn't give me any information about like, well, can they change directions quickly or whatever, right? So again, when we think about validity, did the test measure what it was supposed to measure? Reliability is the reproducibility or consistency of results over several trials. So let's say I wanted to do that caffeine study and I wanted to see if caffeine was going to affect our football players' 40-yard dash times. Okay. So on August 10th, I have, I take 10 of the football players and have them come out. They did their caffeine and they come out to the football field and I measure their 40-yard dash. Okay. It's a nice day that day, right? It's a little bit windy. It's not super humid. It's 75 degrees. Okay. The next day I have the next 10 guys come out. That day it's raining and cold and not great conditions. Okay. Then on the third day, I have 10 more come out. And this day it's 95 degrees and it's hot as shit and there's no wind. Okay. Am I producing consistent results over several trials?
No.
No, because the conditions were different in each scenario, right? So again, if I want my study to be reliable, I have to do everything I can to control those external variables, okay? Objectivity, again, is high reliability with different researchers. Okay, so let's say we wanted to understand body fat percentage and how that affects somebody's 5K time. Okay, so let's say I come in and I measure everybody's body fat percentage and I measure Jelani and I get I don't know, 14% for her body fat percentage. Okay. And then Sia comes in and Sia measures her body fat percentage and she gets 8%. Okay. So two different researchers got two different variables or two different numbers, right? Those are pretty different. 14 and 8 is pretty different. Okay. So we didn't have good reliability amongst ourselves as Sia and me being those researchers in that scenario. Okay. So objectivity would be better. Let's say I got 14% and then Jess came in and Jess got 13.6%. That's pretty good. We had pretty good objectivity amongst the researchers in that example. Okay. Types of tests that we can do, look at me getting little pictures going here. We can do field tests. Field tests are great for estimates. We can use a large group of people. However, we can't control for extrinsic variables. Okay, so maybe I wanted to know, you know, what amount of money people spend on running shoes and how that related to their 5K time in this race, if we ever get to have races again. Okay. So that's good for an estimate, but like we can't control for like, well, that guy got injured and so he didn't really train that much. And this lady just has a good job and she likes to have fancy stuff. So she has expensive shoes, right? So again, we're not able to control for any external variables. However, it's good if I had like 300 people and I could just take how much money they spend on their shoes and see what time they ran in this race. Okay, so that's a decent estimate for us. Okay. A field or lab test, we can use some minimal equipment. It's more accurate than a field test. Here you can see a 3-minute step test. Some of you have done that test before in personal fitness or otherwise. So again, that's a approximate condition. So again, we have some standards we are controlling for some things, but it's not as good as this rigid lab test, which requires us to use sophisticated equipment. Here you can see this chick's got a like a face mask thingy on, right? So we're using rigid controls, the most accurate test here to actually measure what her VO2 max is on a treadmill with the use of this metabolic cart, which tells us how much oxygen you're actually utilizing. So again, in this class, we'll do the best we can, but again, a lot of your stuff is gonna be field or lab tests. We'll get into some of those more sophisticated tests later in your career as you kind of move forward. Okay. When we think about research stages, so first thing we want to do is select a problem. One example you guys talked about was altitude and VO2 max, for example. Okay, so again, I wanted to understand whether or not altitude would help me improve my VO2 max. So first thing I want to do is look into the literature and see what's already been published, what other research projects have been done. Then I'm going to design my experiment based on what's already been done. Okay, so they've done a lot of tests for males, but maybe there haven't been a lot of tests done on females. So maybe we want to focus on that population, for example. Okay, so I'm going to set up my study based on where there's holes in the literature. That I'm going to collect my data, analyze my data via those statistical analyses we talked about, interpret and report my results, and then discuss why we found what we found, which is the most fun part. Okay. One view of research process is this is like a cyclical process. So it starts with curiosity. So I'll give you one example here. So my very first research project that I did was on the menstrual cycle and its effect on running performance. So working with distance runners, and I wanted to know if, you know, where you are in your phase of the menstrual cycle was going to affect how fast or how well you performed. Okay, so that was my problem. I went into the literature. There wasn't a whole lot being done in this particular area. So this was for my master's thesis. So I created my hypothesis. I thought that athletes would perform better in the proliferative phase of the menstrual cycle. I created my research design. I took all the, I asked nicely, I didn't force. I asked all of my athletes, because I was an assistant coach at the time, to participate in this study. So again, I had to test them in each phase of their menstrual cycle to see how their performance was. Okay, so that's what I asked my subjects to do. I collected my data. But what I found was that, you know, we talk about VO2 max being like this measure of aerobic performance. But what I was finding was that my top athletes were not the top VO2 max. Okay, so some of the athletes that were outperforming others in the lab were like my JV girls. And I was like, what the heck is going on with this? Okay, so I found out that the menstrual cycle doesn't really affect performance, but I also found out that VO2 max didn't seem to me to be a very accurate way for me to quantify what their performance was in real life races. So that led me to my next question, right? So what other factors might be important in determining how well these girls are going to perform on race day? Okay, so then my next study looked at body fat percentage. Maybe that was important. Maybe it was running mechanics. So again, cyclical process that allows us to make conclusions, but then maybe draw your interest in another way. So that's my story on that. Any questions on kind of that introductory information? Okay, so other things that I have posted for you for lab 1, you have a copy of Excel, what did I call it? Excel sample. Hang on, let me reshare here. Okay, so this shows you at the bottom exactly how to do each of those tests via Excel, okay? So this shows you repeated measures, correlation, regression, etc. Shows you how to do all this stuff in Excel, okay? Some of you already know how to use SPSS, do SPSS. If that's easier and you wanna do your statistical analysis that way, great. If you don't know how and you wanna use this way is simpler to me, I think, to do than SPSS, but I don't care how you figure out your statistics. You can use either way, okay? So this document is just a guide for you so that you can figure out how to do that. Again, you do not have to do this through Excel. You can do it through SPSS or whatever if you want, okay? Your assignment for today involves, hang on, let me do a new share here. It says copy of lab 1 raw data, I think is what it's called. So there's 3 different data sets, okay? And it gives you some description at the top of what was going on in the test. Okay, so for example, raw data set number 1, they were looking at testosterone levels before and after sleep deprivation. So these men were given 1 week of sleep deprivation. We measured their testosterone before and after, okay? So you're gonna do determine what statistical analysis is appropriate, write a hypothesis, again, make sure it's testable so that we know, how you would measure that. Run your appropriate test, okay? So run the statistical analysis that you determine to be the best one. And then you're also gonna make a graph, okay? How many of you are like savvy in Excel, you know how to do the graphs and that sort of thing? Okay, so some of you are, okay. What would be the best representation of this information? Again, I can see subject 1, I can see his pre and his post. Subject 2, I can see their pre and their post.
A bar graph?
I think a bar graph would be just a delight. So I can go up here to the top, I can go insert charts. If you highlight all of this stuff here, it'll even show you some different things. Wow, look how nice this is. Okay, again, it tells me my pre levels are in blue, my red levels are post, okay? I would need to label this axis, and I can do that through modifying something over here, probably like chart elements, okay? I can add stuff, modify it here. So make sure you label, these are our subject number down here, and these are their testosterone levels in ng per ml, okay? I would want to give this chart a title so that I know what I'm looking at as well. So again, that's just one example there. Okay, so there are 3 different data sets to analyze with different analyses for each one, okay? Any questions on that homework? So really, you're not doing any experiments today. We're just kind of applying that scientific method to some examples. Everybody's okay there? Okay. So then the other thing we should look at today is you have your nervous system stuff on your own tomorrow. So I want to just briefly take a look at that and answer any questions we might have. let's see, neural system homework. Is that what I labeled it as? Okay. Here we go, getting there. Okay. So the first question here, again, this is your first chance to give me a shot with APA formatting and cite your sources. You can choose any neural disease you want to kind of talk about and find 3 different articles to discuss related to such. Okay. Stroop effect is a test you'll do, and then there are 5 reaction time tests that you can do here. The links, I believe, work. They worked a couple weeks ago when I set this up. And then you're going to share your info on that Word or on that Google Doc. Okay. Reflex activity. So there's another handout posted. You'll need the reflex hammer in order to do that. Pretty simple, but then you can also label this reflex arc as well. But the PDF will explain how to do the reflex activities. And then lastly is the caffeine stuff. Okay, so we want to determine how caffeine is going to affect our nervous system functioning. And we need to do some statistical analysis in order to determine whether or not that's true. Okay, so you need to look at least one variable that we are going to measure. So I believe what we need to do as a group is determine what variables it is that we want to measure. Okay, so let me go to the Google Doc. You're gonna let me go? Come on. All right, whatever. So I want you to think about what are some things that we could measure in order to estimate how the nervous system is affected by caffeine. Okay, so I want you guys to think about that. Ready, go. Let's talk about it.
Reaction time.
Okay, so how do we measure reaction time? One of those experiments. Should we do experiment number 2, the stoplight experiment? Okay, and so we're gonna measure, well, I have heart rate already in here, okay? So you're gonna do heart rate. So you'll do your heart rate, then you'll take your caffeine pills, then we'll measure heart rate 30 minutes after, 60 minutes after, 90 minutes after, okay? I gave you all, like, each of the pills that I gave you is 100 milligrams, okay? Take as much or as little as you feel comfortable with. Okay, so 1 cup of coffee is about 100 milligrams. I gave you up to 4. You do not have to take all 4. Take whatever you feel comfortable with for those, okay? Let's think of something else. Get creative here. How are some other ways we could test caffeine effectiveness at home besides heart rate? And we've got this one experiment, this stoplight experiment. Talk amongst yourselves.
Could it be like controlled like exercise or something like that?
What do you mean?
I don't know. Like if it's like for our heart rate, like we could get like our heart rate going up with like a controlled exercise. Or I don't know.
The only thing with that, Janae, and we would do that in real life, it'll be hard for us to standardize that for everybody. So it would be hard for us to know if everybody was like working at the same intensity, just because we don't have like, I'm assuming everyone doesn't have treadmills, you know, or, you know, some sort of way we could like standardize it. So while that is an excellent idea, I don't know if we can do it. What else besides heart rate and relaxing time?
Like our body's temperature.
Okay. Caffeine subjects, do y'all have a temperature thermometer? Okay, I like that. Okay, what else? Let's do one more. One that I always like, and I don't know why I like this, because I like to do it, I think, is the typing test. Again, it's not necessarily reaction time, but it does show like your speed and your error as well. So I think that's kind of cool. Would one of you be able to like right now like search and find a typing test, and then I'll put the link on here for that. Does that sound good?
I'm gonna be the worst at that.
I'm pretty
I
bad.
can't type.
We won't judge you. This is judgment-free zone or something. It's a safe place or whatever I'm supposed to say. Okay, so one of you find a typing test and chat it at me, and I think that'll be good. So you've got typing, body temp, reaction time, heart rate. Okay, and then what I want you to put here where it says amount of caffeine consumed, to put how many, you know, each pill is 100, so you had 200 milligrams or whatever if you had two. And then when you do dosage, I want you to take your body weight and divide that. Okay, so let's say you had 200 milligrams of caffeine, then divide it by your body weight in kilograms. So this will be in milligrams per kg. Okay, so again, that helps us to control for or understand, you know, what you're doing as it relates to body weight. Okay, thank you, Janae. All right, so that link is there for you. Okay. Questions? Questions on today's assignment or what you're doing tomorrow?
And when is today's assignment due?
Tomorrow morning at 8:30.
Okay.
That Excel document.
Okay.
And then the nervous system, I'll be really lenient about when you guys can get that done. So like I said, I'll get the stuff mailed out today, but you know, take some time. Do that, you know, if you are a caffeine subject, you'll want to be caffeine free when you start it. So do it in the morning, you know, do it right when you wake up or whatever, on whatever day. Okay.
I am coming to Dubuque today. If you want me to pick up the package instead of mailing it, you can.
Sweet. Yeah, let's do that. Where are you going to be?
I have to go to the dentist there. So,
Okay.
Yeah.
I'll text you.
okay.
Let's see. So tomorrow you've got your nervous system stuff. Do what you can for now. Hand in your lab number 1. On Wednesday, we will review the nervous system PowerPoint. Okay, there's a couple concepts that I want to go over as a group in class for that. So that'll be good. And then I'll also go over how to do the sensory lab on Wednesday. Okay. All right. Anything else? Any other questions? Again, this will be the longest Zoom session that we have all semester. I promise. All
When's
right.
the next time we're meeting? Wednesday?
Yep. So we'll do Monday, Wednesday, Friday, 8:30. Again, this is the only one that'll be long. I imagine the rest of them will be 40 minutes max. All right. Okay. Let me know if you guys do have questions as you're working on anything. Otherwise, I hope to see you on Wednesday.
Okay. Yeah.